Welcome to this lecture on evolutionary biology. Today, we are going to explore a fascinating concept known as clines. Have you ever wondered how a single species can look completely different depending on where it lives, yet still remain the exact same species? This phenomenon is a beautiful demonstration of evolution happening in real time, right before our eyes. By the end of this video, you will understand exactly how nature balances different evolutionary forces to create these continuous adaptations across physical landscapes. Now that we have introduced our topic, let us start with a simple thought experiment. Imagine you are taking a long hike up a very tall mountain. At the base of the mountain, you notice a specific species of plant that grows quite tall and has broad, flat leaves to catch the abundant sunlight. But as you continue to climb higher, the air gets colder and the wind gets stronger. You notice that this exact same species of plant starts looking different. The plants become progressively shorter and their leaves become smaller and thicker to survive the harsh mountain conditions. This gradual, step-by-step change in how an organism looks or functions as you move across a landscape is exactly what biologists call a cline. Building on that mountain analogy, let us look at the formal scientific definition. In evolutionary biology, a cline is defined as a measurable, gradual change in a phenotypic trait or an allele frequency within a single species over a geographical area. The term was first coined by the evolutionary biologist Julian Huxley in the year 1938 to describe these continuous spatial patterns of biological variation. Unlike a sharp boundary, where one trait suddenly stops and another begins, a cline represents a smooth continuum. This continuum usually correlates with an environmental gradient, such as changes in temperature, altitude, latitude, or moisture across the habitat. Understanding this smooth continuum leads us to a critical question. Why do these plants not just become different species? The answer lies in reproduction. Because the individuals along this environmental gradient can still interbreed with their immediate neighbors, gene flow continues across the entire population. This means that while local populations are adapting to their specific local conditions, like the cold at the mountain peak or the warmth at the base, the continuous mating prevents them from breaking apart into entirely separate species. They remain connected through a chain of interbreeding individuals, acting as a single, highly adaptable biological unit. So why is this continuous gene flow so important for us to study? Understanding clines is absolutely crucial because it allows us to witness natural selection and adaptation happening in real time across a physical space. Without the concept of clines, we might mistakenly look at the tall plants at the bottom of the mountain and the short plants at the top and incorrectly classify them as two completely different species. Furthermore, clines help conservationists and ecologists understand how species might respond to shifting environments, such as global climate change, by tracking how these trait gradients move over time. This brings us to the underlying mechanics of how a cline actually forms and maintains its shape. To truly understand this, we have to look at the forces creating it. A cline is typically maintained by a tug-of-war between two evolutionary forces, natural selection and migration, which is also known as gene flow. Natural selection tries to make the cline very steep by perfectly adapting local populations to their specific environments. On the other hand, migration tries to flatten the cline by mixing genes across the landscape. The ultimate width of the cline depends entirely on which of these two forces is pulling harder. Now that we understand the tug of war between selection and migration, let us look at how evolutionary biologists calculate this mathematically. The formula for the width of a cline is W equals 2.4 times sigma, all divided by the square root of S. Let us break down these symbols. The variable W represents the width of the cline, usually measured in kilometers or meters. The symbol sigma represents the dispersal distance, which is the average distance an individual organism travels from its birthplace to where it reproduces. The variable S represents the selection coefficient, which measures how strongly the environment is favoring or punishing a specific trait. The number 2.4 is a mathematical constant. If dispersal distance increases, the cline width increases because animals mix genes widely. Conversely, if selection pressure increases, the cline becomes narrower and steeper. Before we move on, let us see this formula in action with a concrete example. Imagine a population of wild mice living across a landscape that transitions from dark volcanic rock to light-colored sand. We are given that the average mouse disperses a distance of 5 kilometers, so sigma is 5. The selection pressure against the wrong coat color is quite strong, with a selection coefficient of 0.16. We need to calculate the expected width of this coat color cline. First, we take the square root of the selection coefficient, which gives us 0.4. Next, we multiply the constant 2.4 by the dispersal distance of 5, giving us 12 in the numerator. Finally, dividing 12 by 0.4 gives us a final cline width of 30 kilometers. This means the transition zone, where we see a mix of dark and light mice, spans exactly 30 kilometers. Now that we've successfully used the formula to find the width, let us look at a different scenario where we work backward to find the selection coefficient. This is a crucial derivation biologists use when they can observe the cline but do not know the environmental pressure. Imagine a species of pine tree growing along a mountain slope. Biologists observe the width of the freezing tolerance cline to be 12 kilometers, and the seed dispersal distance is 2 kilometers. We start with our standard cline width formula and substitute our known values. The width, W, becomes 12, and the dispersal distance, sigma, becomes 2. We simplify the numerator by multiplying 2.4 by 2, which gives us 4.8. Our equation now states that 12 equals 4.8 divided by the square root of S. Continuing with our derivation, our goal is to isolate the unknown variable, the selection coefficient S. To do this, we need to get the square root of S out of the denominator. We multiply both sides of the equation by the square root of S, and then we divide both sides by 12. This algebraic rearrangement sets up the equation so that the square root of S is equal to 4.8 divided by 12. When we perform this straightforward division, we get a value of 0.4. This 0.4 represents the scaled strength of natural selection, but it is still under the square root. We are now just one step away from finding our final selection coefficient. This brings us to the final step of our derivation. To find the actual selection coefficient S, we must eliminate the square root. We do this by squaring both sides of the equation. Squaring 0.4 gives us a final selection coefficient of 0.16. What does this number actually mean in the real world? It tells us that there is a 16% fitness disadvantage for pine trees that have the wrong freezing tolerance for their specific elevation on the mountain. By working backward through the math, we have successfully quantified the invisible force of natural selection acting on this population. Now that we have calculated the width and selection pressure, let us look at how this actually appears on a graph. Here you are seeing a classic representation of a cline. The horizontal axis represents the geographic distance in kilometers, perhaps moving up a mountain or across a continent. The vertical axis represents the allele frequency, ranging from 0 to 1. Notice the smooth S-shaped curve. It starts high, meaning one trait is dominant in that region. As we move across the landscape, the curve drops steeply. That steep middle section, marked by the double-headed arrow, is the Cline width we just calculated. It visually represents the transition zone where the two evolutionary forces of migration and selection are actively battling. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that a cline is a beautiful demonstration of evolution, representing a gradual change in a species' traits across a physical landscape. Second, we saw that continuous gene flow is the glue that holds the population together, preventing it from splitting into different species. Finally, we explored the mathematics behind this phenomenon, learning that the width of a cline is a delicate balance between migration spreading genes and natural selection filtering them. You now have a solid understanding of how nature dynamically adapts to changing environments. Great job following along with the math and concepts today.